The purpose of my research is to try to improve the quality of life of individuals with Down syndrome through research. I've been doing this research for 19 years now. I have a daughter with Down syndrome. Her name is Tishi, and she was born in 1995, so in June 1995. So in about a year, I basically had to retrain myself, repurpose everything that I had learned before, and basically try very hard to become a Down syndrome researcher, a person who could actually one day try to help people who share the same disorder as my daughter. So, I'm a physician by training. I'm a scientist by profession, and I'm a father. So what it helps me do is to basically see Down syndrome in 3 different ways. And just like when you put together a 3-dimensional picture, sometimes you need like 2 or 3 points of view to actually see the whole. And I think that helps me a lot, because I get to see the person with Down syndrome as a person first, as a human individual. I get to see the person with Down syndrome as a person who has a disorder that affects the nervous system and parts of your body in many different ways. So that's the physician part. And I get to see a person with Down syndrome as somebody who has this genetic disorder called Trisomie of chromosome 21 that affects different systems in your body, including the nervous system. And because of that, I get to see different aspects, and each one of those aspects, each one of those levels, they talk to each other. Over the course of those 19 years, we have found interesting things, and having my daughter actually has really made me attuned to a lot of aspects that I wouldn't have thought before. In the beginning of my career, for example, I start looking at motor function in mouse models of animal models of Down syndrome. Most people were just thinking of memory and learning and all those things, and I was thinking of motor function and motor development, which is very critical for the development of many functions in societal living. You know, it's just one of those things that we take for granted. As a kid is playing with another kid and they're both running around, and all of a sudden you look at your daughter, she can't run as well, or she can't run as fast, she can't climb stairs as well. And wow, I mean, if we're trying to model this with an animal, the animal has to model that too. So that was the beginning of a long process. We found out some things related to eye movement in folks with Down syndrome. So that's when I start transitioning a little bit in terms of looking into function, neurological function in actual human beings with Down syndrome. And we found out that some reflexes that actually keep your balance, again, going back to that whole theme of motor function, some of those reflexes that keep your balance, they're very depressed in individuals with Down syndrome. And that was the first time that anybody had shown anything like that. And then when I was in a medical center where we could start actually doing clinical trials, we followed some of the preclinical work that we have done. So first, the first drug that we actually tried in an animal model of Down syndrome was fluoxetine. That's now is also known as Prozac, that now people are actually putting into clinical trials. And we started it all. That was the very first paper where anything in terms of a drug changed anything in the central nervous system of a mouse model for Down syndrome. And it actually pretty much rescue or eliminated the bad effect of the trisomy, in this case in the mouse, of this piece of chromosome 16. with a single drug, we saw that we could do something, and that really encouraged us. And then we moved forward with another drug called memantine. Memantine is an approved drug for Alzheimer's disease. And because it was already approved, it's approved both in the European Union and in the United States, been approved here for much longer, and that had a very good safety profile. I approached the FDA and I said, here, we have this preclinical data with the mice. There is this connection between Alzheimer's and Down syndrome. This is an approved drug for Alzheimer's. Can I test that drug in a few individuals, a small group of individuals with Down syndrome? And then the first clinical trial that was based on studies that were done in a mouse was born. We did this pilot trial with 20 people in one side, 20 people on the other side. Half took the memantine, the actual medication. The other half took a placebo, a sugar pill. And we compared the effect of those. And even though the effects were quite mild, and we were only able to detect the improvement in one measure of cognition, that was the first time that anybody had seen anything like that. And but the important thing of the data is what the data didn't accomplish, which is we saw some smaller effects that statistics told us that if we did a study that was a little bit bigger, maybe 2-3 times bigger, we would be able to actually see effects in 5 different measures of cognition. And that was a short treatment. It's 4 months of treatment. We were able to see quite a few things, that some of them that we were not expecting to see in terms of potential improvement. So we're now following up that trial with a larger phase 2 clinical trial involving 200 people with Down syndrome. It will be about 2 years before we actually see results for that, but this is the long game. They're basically tissue that's collected from adults with Down syndrome. We actually are trying some innovative methods to obtain those cells. We're collecting cells from urine samples, different from other folks who are actually doing real tissue biopsy, like skin biopsies, which are mildly invasive, but they're invasive nevertheless. And with that study, we have been able so far to differentiate those stem cells that we make from renal epithelial cells derived from your kidney. We are able to transform those cells into a pluripotent state, a state very similar to being a stem cell. And from that point, you can differentiate them into different types of tissue. So far, we've been able to differentiate those cells into glutamatergic neurons, which are like one type of excitatory neuron in the brain. And we have been able to derive them into cardiomyocytes, or heart-beating cells. It means a lot. The first beacon of hope for anything to happen was when I started reading Dr. Lejeune's work. I mean, Dr. Lejeune was very influential for me to become the person who I am today. Sees the individual with Down syndrome as the human person all the time. So he was very influential to me. I feel like a little bit empty because I'd love to be able to thank Dr. Lejeune for everything that he has done, because he has made a huge difference for this field. It is a great day because it means a lot to me. I mean, it really does. I mean, it brings me that much closer to the man who started it all. So.
Thank you very much.